Hello, my name is Chris, and welcome to this short tutorial on proteomics quality control, which is our software to create comprehensive quality control reports. Now, pdxqc was originally developed to analyze the output of MaxQuant, which is a popular software package to process your typical shotgun bottom-up proteomics raw files. In the meantime, our software also supports other software packages, such as OpenMS, although to a lesser extent. So for the next 10 minutes or so, we'll motivate how QC can actually help you save time and make your life easier. And we'll explore some of the features of pdxqc. First, we'll motivate why QC is important and what kind of QC we're actually talking about. Then I'll show you how to install pdxqc and then create reports using a very simple drag and drop mechanism without actually writing a single line of code. And finally, we'll explore how a report looks like and find some interesting use cases. So let's get started. Here we have a typical workflow of a proteomics project. You hopefully first plan your experimental design, maybe do some preliminary studies, optimize methods. Then you go on collecting your samples, preparing the samples before you can measure them on the ASMS system. Yeah. Or maybe you skipped all of those steps and you just downloaded some raw files from some internet archive, Proteome Exchange, one of its members, for example. Yeah. Once you've measured or obtained the data, you plug in the raw data into some kind of bioinformatics software, so MaxQuant, OpenMS, TPP, you name it, which will do the data processing. In order to do that efficiently, you probably have to tune the software parameters to fit your instrument, your sample type, and then you pray to God it doesn't crash. Now, in case it didn't crash, before you move on to data interpretation, it is highly recommended to check data quality. Yeah? So to perform some kind of quality control or quality assessment, now there are specialized tools out there which can help you monitor, for example, your instrument performance using a known calibration mix. But this is not exactly what we're talking about here. We assume you already did that. However, you should also in parallel make sure that the samples that you measured are actually okay as well, right? Because not all problems can be detected by regularly measuring calibration mixtures. For example, your digestion might have failed, right? You have insufficient material on the column, or you're simply using the wrong software settings, right? All of this could go wrong, so you need to check. Yeah? And once that check came out fine, you can go on to do data analysis. So some pathway mapping, Go enrichment, yeah, you name it. Now, we will concentrate on the section highlighted in red in more detail now. Starting with the sample on the left, yeah, which may or may not be metabolically labeled, we typically digest proteins to peptides using trypsin and perform a complexity reduction step using some kind of separation technique, HPLC comes to mind. And then we measure the sample over the course of minutes to hours on a mass spectrometer, which then spits out some kind of raw data, so many spectra in some vendor-specific format. So this will contain lots of MS1 and probably even more MS2 spectra. We then process the data, for example, using MaxQuant, and what happens next is typically very much dependent on your individual preference. But many people, if they don't know about quality control software, will start to look at the data in a manual fashion, do a data check. So in the case of MaxQuant, you would probably go and open up the summary TXT file in Excel and inspect some numbers, right? Number of peptides, for example. Then you move on to protein groups TXT and see if your protein of interest shows up. Similar things can not only be done in Excel, but with Perseus, which is MaxQuant's interactive data manipulation program. But this is all very manual, right? Now, typically, you follow those steps for every study you're doing, and it gets repetitive and it's boring, and maybe you forgot to check something. So now, on the rare occasion, the data, if it looks bad, for example, the number of identified PSMs is low, or the number of proteins is low, you start digging, right? And you ask yourself, why is that? Did, for example, the digestion fail? So at what stage did it go wrong? Did I inject enough sample? Did I configure the FASTA file correctly? And so then the question is, what are good metrics which correlates with some of these sources of failure. And so you quickly come up with a set of metrics which you think are useful and which have the power to discern at what stage actually, where did it go wrong. And so this is exactly what pdxqc is doing. Luckily, MaxQuant provides a set of output tables in TXT format, which can be used to check and visualize and even automatically score at what point your experiment failed. Yeah. So, for example, to judge if cyclic labeling works, you can look at label incorporation. Yeah. For digestion efficiency, we can look at the number of miscleavages, where the majority of peptides hopefully does have none. Yeah. You can look at the peak width of the chromatography, at the alignment if you're comparing multiple samples. And so some parameters which might also tell us how to optimize the method. So for example, in order to maximize the number of PSMs, for example, we could look at oversampling, which is the number of times a single peptide was repeatedly fragmented for MS2. And finally, you can, for example, look at peptide and overall protein counts. And if those are fine, it typically means all the rest is fine. If those are not fine, it probably means one or a few things along the line went wrong. So we can not only do QC, but we can also do optimization of the method. Yeah. Now, some of these metrics you will quickly find are interdependent. So for example, if you have a low identification rate, this usually correlates with low peptide intensity and or some contaminants, high amount of contaminants. If you have a very high peak width in LC, this usually means if you didn't adapt your settings, usually means you have high oversampling of MSMS events. All right. So by computing all of these metrics, we can now also automatically decide very quickly if something failed and where that happened. Sometimes the problem can be fixed by simply adjusting some parameters in MaxQuant. Sometimes, so if you have a problem, the issue is more upstream and you need to decide if it's worth to rerun the sample, if you can. So how does it work in practice to work with pdxqc? Well, after you've run MaxQuant on your raw files, you will find that MaxQuant created a subfolder on your local hard drive, which is called txt. This is the output folder of MaxQuant, and the data in there we're going to use to obtain a report. So if you look into this folder, pdxqc is going to inspect a subset of files in there and create certain metrics and plots. And it will, from this, generate a PDF or HTML report, and in addition, some other auxiliary files. For example, an MCQC file which stores QC information in a machine-readable way. In some cases, you do not have access to the full TXT output of MaxQuant, but only partial data. For example, if you downloaded partial data from the internet. If some of those files are not present, it's no problem. It just means that the metrics which are derived from those files cannot be computed. The most important file at the moment is evidence.txt. This is where most of the metrics are derived from. So if you have that, you get a pretty comprehensive report already. Okay, so let's get to installing pdxqc, and then we'll look at some examples. The installation instructions, detailed installation instructions, can be found at our basically website on GitHub. And in short, we'll need two essential ingredients to run pdxqc. Since pdxqc is an R package, we'll need R, and to allow the creation of HTML reports, we need another software called pandoc, which is a universal markup converter. And this will be invoked from inside pdxqc, so don't worry about it. You just need to make sure it's present. So now let's dive into the installation, shall we?
So let's have a look at how to install pdxqc. I pulled up our main web page. Let's look at drag and drop installation. So there's a vignette for this. We're just gonna first download R. So this is a vanilla virtual machine, it doesn't have anything, so let's just download R. You can skip this step in case you already installed a recent version of R. Just make sure it is a 64-bit version. These days, if you install these things, both 32 and 64-bit, come in the installer. I just deselected 32-bit because you don't want to risk running 32-bit just by accident. Okay, so that was R done, installed. Second thing we need is pandoc, which I'll just open the browser tab for this. We're not going to download it yet because we'll just do that later. For now, let's open up R. So that's the R console. You can use any GUI you want, basically RStudio, whatever you like. I just used the plain vanilla R one. Let's install pdxqc, select the mirror. Okay, it's going to do its thing. pdxqc installed, very nice. And the last thing we need to know is where, in what library in our terminology, was pdxqc installed. And you can see here it's in the R main folder, so that's okay. So there's no dedicated extra library we need to worry about. This could be the case for your installation. So now let's just go to where R was installed. Particularly, let's go into the library folder and find pdxqc. Okay, there we go. We need the drag and drop folder because we're going to copy that to any place you basically like. I just picked the desktop, and now we're going to copy R, so the whole R installation, into a subfolder of this drag and drop. So it's a little bit cat and mouse here. If you read the installation instructions, it tells you exactly what to do. So basically, we need the main folder of the R installation, copy it and paste it into the internal subfolder of our drag and drop feature, basically. I'm going to copy that. Okay, sweet. Now we don't need the original R installation anymore. You could even uninstall R at this point if you wanted to. We have everything we need. Now let's worry about pandoc. Download the zip file. So it's not an installer, it's just a plain zip file which contains one big pandoc executable, which is in here. We're going to copy that and we're going to paste it into the bin subfolder of our local R. And you need to create a subfolder called pandoc. We're just going to put the pandoc executable in there. Okay, nice, done. Now there's one last thing we need to do. This batch file, which is going to be our main target, which we're going to use to enable the drag and drop feature. By default, it assumes you installed R 3.1.0. Well, because it cannot know what kind of R you installed. So just take the actual name of the R installation that we just copied into our internal folder and replace it. So here we used 4.1.1. So we're going to save the batch file using our local setup, and then, well, it's ready to use. So we need to drag something onto this batch file, and here I have a txt folder. I just drag it onto there, and the batch file is going to get invoked, and it's going to run pdxqc, and it will create a report and all the other auxiliary files within that txt folder. Now we can go fancy and create a shortcut to this batch file. Put that anywhere you like, so you don't have to open up this subfolder anymore. And now you can just drag and drop onto the shortcut, and basically the same thing will happen. Okay. So now if you look into the txt folder, you can see the report was generated. Yeah, and that's pretty much it.
So, welcome back. Let's put the drag and drop feature of pdxqc into action and inspect some reports. So what I have here is a maxquant run which just finished, and the folder on which maxquant run is this yeast folder here. So let's quickly dive into that. Here we have the raw files, 2 of them which maxquant operated on, and all the other files that you can see are basically intermediate and final result files of maxquant. One file to mention here is the mqpath file, which contains all the parameter settings that we configured maxquant with. And this is a file that pdxqc will inspect in order to derive thresholds for certain plots. Now you can leave that file here, or even better, you can copy it to the txt folder. So I'm just going to copy the file, and then now we're going to look at the txt folder, which is here under combine txt. Those are the files now which pdxqc is going to read and derive the metrics from. So we're just going to paste the mqpath file here just to make sure those files now belong together. We could go even fancier and copy the txt folder somewhere else just to make sure that they stay in sync. We're not going to do that for now. Now let's generate the report, which we can simply do by dragging any of these files onto our batch file, and then this neat little window here is going to pop up. pdxqc is going to run. So now let's fast forward till the end and let's look at the result. So welcome back. pdxqc finished in just under a minute. So we can close this window, and now you can see some additional files which were created by pdxqc. The most important one is probably the HTML report, which is a rather large file because it contains some images which are embedded into the HTML. Then there's a log file, which basically is a copy of all the output of the window that we just saw. So in case you want to look up what pdxqc told us there, you can do that here. Then there's an mzqc file, which contains machine-readable, basically metric data. Then there's a PDF file, which is basically telling you the same story as the HTML. It's just a bit less interactive. Then we have a configuration file, and we're going to inspect that when we look at the HTML report. And then there is another file which we're also going to see when we look at the report. Yeah, so that's basically it. So let's now jump into the report and have a look at it. So here I have the report pulled up, and the first thing you're going to see is basically an index which allows you to quickly jump to any part of the report, in particular to any of the metrics you can see here. So for this particular set of data, pdxqc generated 35 metrics. The number of metrics you see for each dataset depends on the type of dataset. So in this case we have 35. Now let's quickly jump to the first section, which is overview. So here you can see a neat little feature of pdxqc. If you run maxquant in a non-English locale, meaning where dot is not the decimal separator, maxquant will generate bogus results. Usually the number of PSMs will be very low, and maxquant won't tell you that something went wrong. So pdxqc in this case will detect this problem and it will not generate a report. So the very fact that we can see a report means the maxquant run was done using an English locale. The second thing that every report will show you is what is the system that I'm running this browser on. Is it correctly configured? Meaning, does it have an English locale, and could I run maxquant on this? And the answer here is yes, you can. If at some point this turns red, do not run maxquant on this very system where you're executing the browser on, or change your locale first before running maxquant, because otherwise the maxquant results will not be useful. Okay. So next up we have a quick guide, which we're just going to briefly go over. You can collapse sections by clicking the name, okay, if you want to get rid of it. Then, very important, each metric, which are down below, has a show help button, so you can basically read on what the metric is trying to tell you. And this is available for all the metrics, so you can quickly read up on what you're supposed to see and how to interpret this. It will also tell you how it scores this metric and how that's done. And we'll come back to that in a second. Okay, so that's available. Going back, you have a config file, which is right next to the report. We talked about this earlier. So you can look at this config file, and this is basically how pdxqc was configured, and we just used the default configuration. If for some reason you want to change that, For example, you could say, well, I only want an HTML report, I don't want a PDF report, you just delete the line here and save some time. Or you can set individual thresholds, for example, for the total number of proteins you expect per sample. This is something that pdxqc cannot know because it depends on the sample type and your settings and your instrument. So this is something you can set, and this will influence the scoring of some of the metrics. Most metrics have a scoring scheme which is independent of magical thresholds, but there are some, right, which you can set here. You can also enable or disable certain metrics if you find them, not useful for your scenario. All of this, given that now once you change the file using any text editor of your choice, you need to rerun the report generation, and then this config file will basically kick in. All right, let's go back to the report. What else do we have? We have a file name sorting txt, which is also right next to the report. So this is basically the attempt of pdxqc to make raw file names in the plots more readable, because those names can get pretty long, which means the axis labels are also going to get pretty long. So what pdxqc is doing, it's going to find common substrings in those names and remove them. So this is basically the subtext that remains, and those are the raw file names which pdxqc came up with, and this is what it's going to use. If you're not happy with those, you can just edit the text file and use whatever name you like. Getting back to the report, you could even, if you wanted to, edit the markdown template for this HTML report and customize the hell out of it, but we're not going to go there for now. Okay, then if you find a bug, something that you're not happy with, there's a direct link to the issue tracker on our GitHub homepage, so you can file an issue there, or if you want to have a look at the full manual, it's also linked here. Okay, now let's look at the actual data that pdxqc produced. The first thing that you're always going to want to inspect is the heat map. The heat map consists of all the metrics which are automatically scoreable by pdxqc, and on the y-axis you're going to see all the raw files that MaxQuant processed. The heat map is color-coded. If you see a green tile, that means very nice. You always want to see green. If you're seeing black, that means, well, this metric is underperforming and might need further inspection. If you see it red, that means, well, something went very, very wrong, and you should definitely look at this. Some of those metrics depend on magical thresholds. For example, we have a peptide count metric where we expect per raw file like 15,000 identified peptides after a false discovery rate. This very much depends on your setting, obviously, and in this case for the yeast data set this default might be a bit too high. That's why it doesn't really score very nicely. So you can change that and rerun the report. So that's the case here also for the protein count or for the expected peptide intensity. But looking at this heat map, we can already see those two files, which were supposedly to be repeated measurements of the same sample, they didn't quite yield the same results. The file here definitely has some problem, and now we need to figure out what it is. So as I mentioned earlier, some of the metrics are pendant. So we can, for example, see that the peptide intensity of all the identified peptides is worse in the second sample than it is in the first. Also, you see that the top N MS2 spectra that the instrument decided to do is not very nice. So there's probably potential for optimization here, but that might not be our primary problem. What we can see here is the mask calibration before MaxQuant corrected it. And this is the corrected calibration values after MaxQuant did an in silico correction, basically. So this seemed to work very nicely for one file, but it didn't work for the other. And this might actually be the reason why we see bad peptide intensities, because if the calibration failed, all the peptides that we identified are probably wrong anyways. You can also see that in the identification rate for all the MS2 spectra, it's, well, okay-ish for this sample here, and it's really bad for the other. So all of this might have to do with our mask recalibration not working properly. So let's quickly jump to that section and we can use the index here above. So there's an uncalibrated mass error. So let's look at this. You can also have a look at the help here. Now we're going to quickly see this is our first raw file, and MaxQuant determined using a wide tolerance basically that it was decalibrated like 18 ppm. You see those thresholds here, which pdxqc derived from the mqpar file, and MaxQuant would only search plus minus 20 ppm in the first place, which means that if your sample is decalibrated even worse, it might not catch that. And so even though you're seeing for the second sample that it seems that all the masses are somewhat nicely calibrated in the middle, pdxqc looked at this data and said, well, that's probably not correct, and it labeled those as out of search tolerance. And it did that based on the standard deviation, meaning here the width of the box, which you would expect to be much, much smaller. So for the sample where we just had an accuracy problem, the standard deviation is 1.1 ppm, whereas for the sample which looks weird, it's 5.6. So something is definitely wrong here, and pdxqc basically tells you you need to search a wider window using MaxQuant. Now, if you look at what MaxQuant did using this, correction that didn't really work, is it was able to correctly calibrate the first sample, which you can see here. It's nicely centered around 0. But for the second sample, again, pdxqc tells you it's probably out of search tolerance. And also the height of the bar tells you how many peptides were actually identified, and you can see that, well, it's not really great. What we should do now is basically rerun MaxQuant using a wider search window. Here I generated another report after running MaxQuant again using a wider search tolerance for the MS2 spectrum. So let's quickly check the heat map, and this one looks much nicer. Maybe I can do some post-editing magic here and show you the other heat map just for comparison. Let's look at the decalibrated mass error again, which is somewhere here, right? And you can see I selected a different threshold. If you want to know exactly what, I selected 50, right? So this help is a bit interactive so that it adapts whatever search parameter you used. So I chose 50, which should be the lines that you see here. And now all of a sudden, the second sample could be successfully recalibrated by MaxQuant. The standard deviations are also down, which is nice. And so we have a lot more peptides identified. And now those samples are actually comparable. Okay, so what else did we see in the first report, which might trip us a bit? So again, or still, let's say the top and high is bad. So let's see why that is. Top N. So yeah, here you can basically see the problem. This is an optimization problem. It's not really that something wrong with the data, but maybe you can optimize your method a bit. This is basically the number of fragment spectra that the instrument triggered after each full scan, and you can see it pretty much drops off very quickly after, let's say, 7 or 8. Configured was up to 20, I think, in this case, and you can see that it might be worth for this kind of sample to cut it down to 10 or shorten your gradient, whatever you decide to do. But doing 20 looks like a waste of time. That's that for this sample. I have another sample prepared, which has a different problem, so let's look at that one. Welcome back. I pulled up another report, which is from a human QC sample, which was actually also used in the publication script of pdxqc. So here, if we inspect the heat map, we can see that we used 5 raw files. Here you can see the metrics, and there's something going on here. Again, top and high is not really what it should be, but that's nothing we can change after the fact. We just know we can maybe do better next time. But something is wrong here in the match between runs metric. So let's quickly look at this and see what's wrong there. Where do we have it? Here, match between runs. So what this basically tells us, MaxQuant was instructed in case it could quantify a peptide intensity signal in MS1, but it could not maybe identify it because it was missing a corresponding MS2 spectrum, or it couldn't identify what the spectrum was after false discovery rates filtering. So what MaxQuant can do is, based on an accurate mass and time tag basically, it can transfer a peptide spectrum match from one raw file to the next. What you need for that is obviously comparable retention times. So MaxQuant will try to correct and align retention times across raw files. And this metric basically tells you how good this worked. So what you can see on the x-axis is, well, the retention time of each of those raw files. You'll see that the first file is missing because that's going to be our reference. And the y-axis tells you, well, compared to the reference, how far are you off in actually aligning peptides which should have identical retention times. So ideally, you want to see all those dots, which each represents one peptide. You want to see those on a single line here, right? They should all be green. So they're colored if they're basically within a delta RT of 1 minute in this case, because that's what the MQPAR file tells us how the matching was done. So if you're within 1 minute, you're basically green, and you get a score, in this case, of 100%, because give or take 100% of all these peptides are aligned perfectly to our reference. Now, that's the case for the second file. For the third file, it doesn't really look that nicely. Apparently 73% of all the dots are green, the rest is red, so questionable. But then in file 4 and 5 it gets even worse. We can see something which is intriguing. So what you see here, basically the delta compared to our reference for identical peptides basically is up to, what is this here, 30 minutes. Yeah? And now if you know exactly how MaxQuant default settings are working, MaxQuant only searches up to 20 minutes to derive a retention time correction. And basically what MaxQuant did to correct the retention time is shown in blue here. So basically if I pull on this blue line and I pull it upwards, I would also pull upwards this corresponding line here. So in order to pull this to 0, I would need to pull this line up another 30 minutes, which means I'm already at 10, so I would probably end up at plus 40 minutes, which MaxQuant is not allowed to do according to its default settings. So basically it never had a chance to correct this retention time discrepancy. Now, given this is quite an extreme sample, but if you measure a large cohort on a single column, this might be something that would happen to you. What do we do about it? Well, we run MaxQuant again, but just with a larger threshold to allow it to correct larger retention time differences. And this is basically what I did here. So I changed MaxQuant parameters, ran it again, and if we now look at the alignment, and I think I allowed 100 minutes, which is really extreme, but MaxQuant now should be able to correct the retention time of all our raw files and align them perfectly. And you can see for file number 4, it corrected, it said, well, I need to correct peptides up to 40 minutes in time. And now it can align some of the peptides, 38% of them, but for whatever reason, MaxQuant fails to align especially the first and the last part of the gradient, which means that it's still not perfect. So you might question if you want to leave this match between runs features enabled anyways, because it might do more harm than good. And the question is now, how much harm does it actually do to transfer identifications which are apparently off, by more than a minute or so? And the answer is right here in the next metric. So you can basically judge how severe the problem is, and so what is the gain and what are the potential false positives that you're including here, which will obviously have an influence later on your peptide and protein quantification. So what you see here is you don't want to see red, basically, right? And so this metric says, well, if you do match between runs correctly, you should only see a certain peptide in a certain charge state. You should only see it once per raw file. If you see it multiple times with a large delta RT, this basically means that one of those two identifications is probably wrong. And if you just look at the local identifications, which are here called genuine, of that raw file, you see this happens very, very rarely. Now, if you just look at the transferred identifications, you can already see that happens quite a bit. And all those identifications, so let's say 10% of them, are definitely false, right? Because you see the same peptide twice in the same raw file at vastly different retention times. So whatever quantification you derive from those, they're going to be off, right? And so if you take both these informations together, this is what you end up with, and you basically need to decide if it's worth doing match between runs or just switching it off and just live with fewer peptide spectrum matches. How many exactly you get, you can see here. So basically, if you look at peptides and proteins, the green stuff is what is generally available within the raw file anyways, so without match between runs. Then there's an overlap of PSMs which are generally present and which have also been transferred, and those might actually be the problematic ones. And then this is basically the blue stuff is what you gain by transferring PSMs across raw files. So this is the gain that you're potentially losing if you switch off match between runs, and now you need to basically decide if it's worth it or not. Okay, so that's basically a short run of our report. I hope you found this useful, and now let's go back and discuss some final details.
So now, after we inspected the output of pdxqc, I'll leave you with some further reading if you want to, right? So there's the original paper, which we published some years ago. If you want more information, you can look at our GitHub homepage, basically. If you have questions, want to report bugs, or request new features, your contributions are always welcome. So remember that I told you how to install pdxqc. Now I'm going to tell you there's a much easier method to do it. You can just visit our web servers, where we basically run an RShiny application, which basically allows you to upload your MaxQuant data and obtain a result just using a web service. So if you want to play with pdxqc without the hassle of installing it, just go there and try it out. Future plans are to complete our MZQC implementation, which currently only reports a few metrics. We're hoping to extend that so that basically everything that you see in the HTML or PDF reports is actually also showing up in the MZQC file. Then we hope to extend the support of OpenMS, which basically currently creates the evidence file, but not all the other files. So you have a limited set of metrics already, but we hope to extend that to the full set, basically. And then, since MaxQuant released version 2.0, MaxQuant also supports swath data, basically. And currently, pdxqc does not know how to deal with this kind of output. So we have to first come up with some new interesting metrics for swath data, and then actually implement some of those changes. So that's something that will come up in the future. Let me close by acknowledging my former colleagues at MDC Berlin, where we initially conceived pdxqc, and without whom this project would not have been possible. Many people, including many of my students, have worked on pdxqc over the years, and I also invite you to share your ideas, metrics, improvements, or even code contributions. So if you have questions, comments, or feedback, please feel free to contact me via GitHub or email. Thank you.